Dear students, today we are going to study endoplasmic reticulum, which is a very important organelle of a cell. It is a membrane-bound organelle. It was first of all discovered by Porter, Claude, and Fuller, and found in all the cells of eukaryotes excepting RBC. But it is wanting in prokaryotes because they do not have membrane-bound organelles. They are highly organized in cells engaged in protein synthesis and lipid metabolism. If you wish to identify the endoplasmic reticulum membrane, the marker enzyme is glucose 6-phosphatase. Now, if endoplasmic reticulum is present in the cell, it divides the existing cytoplasm into two, that which is enclosed in endoplasmic reticulum and that which exists outside the endoplasmic reticulum. It has this distinction has certain significance, that in the membrane bound cytoplasm the enzymes they get localized. It is very important for maintenance of ionic and pH gradient. It is helpful in creating diffusion barriers because all the enzymes they are not required every time, and diffusion barriers are very important for the movements also, and restriction of movements of certain ions. Creation of potential differences in the membrane is effected by presence of endomembrane systems. The whole of the cytoplasmic reticular system has three components. One is endoplasmic reticulum, another is nuclear membrane, then Golgi complex. Now we have two types of endoplasmic reticulum. One is smooth endoplasmic reticulum and another is rough endoplasmic reticulum. The smooth endoplasmic reticulum is also called as SER. They have smooth membrane because they are devoid of ribosomes. That means ribosomes are not present on such kind of endoplasmic reticulums, and this absence of ribosomes give them a smooth appearance, and they are majorly indulged in synthesis of steroids, and they are majorly found in cells which carry out glycogen metabolism and lipid synthesis. For instance, adipose cells, glycogen storing cells of liver, and leukocyte, etc. The smooth endoplasmic reticulum, they are involved in a very vital cellular phenomena, that is cellular detoxification. Now we have another kind of endoplasmic reticulum, which is rough endoplasmic reticulum and is also called as RER. Many a times it is called as ergastoplasm also. They are termed so because they have presence of ribosome all around them, and that imparts them a peculiar characteristics of looking rough. These endoplasmic reticulums, they are involved in protein synthesis. So we students, we have seen there are two kinds of endoplasmic reticulum. One is smooth, which is involved in cellular detoxification and steroid synthesis and lipid metabolism. Another is involved in protein synthesis because it is hugely laced by number of ribosomes, and ribosomes, as you all know, that they are involved in protein synthesis. Now come to the structural organization of the endoplasmic reticulum. Endoplasmic reticulum exists in three forms. One is vesicles. Vesicles are actually rounded, spherical, or ovoid. They are predominantly present in cells which are indulged in synthesis of proteins, for example, the cells of liver and pancreas. Their dimension is around 25 to 500 microns. Another physical form in which the endoplasmic reticulum exists are cisternae. Cisternae, as you can view in the slide, are unbranched, elongated, flattened sac-like, present in the cells which are busy in synthesizing proteins. For instance, the cells of pancreas and brain, they have huge number of cisternae enclosed in their cells. They are around 40 to 15 microns in dimension. The third form in which the endoplasmic reticulum exists are tubules. Tubules are branched, forming reticular system found in the cells, and they are present in the cells which are busy in steroid synthesis and around 50 to 200 microns in dimension. So these are the three structures: vesicles, which are rounded, spherical, or ovoid; cisternae are elongated, flattened sacs; protein synthesis, present in protein cells busy in protein synthesis, whereas tubules are found in cells which are majorly involved in steroid synthesis. Come to the ultrastructure of the endoplasmic reticulum. The membrane of endoplasmic reticulum is thinner than the plasma membrane, and also its fluidity is lesser than that the membrane of the plasma membrane. Its membrane is a double unit membrane as of plasma membrane, and its thickness is around 4.5 to 5.5 nanometer. So it is not hugely different from plasma membrane. It is very much similar to plasma membrane, but somewhat it is thinner, and its fluidity is lesser than that of plasma membrane. Then come to the chemical nature. You would be able to come across following biomolecules in the membrane of endoplasmic reticulum. For instance, glucose 6-phosphatase, nucleotide diphosphatase, NADH cytochrome C reductase, NADH diphosphorase, sterase, and magnesium activated ATPase are generally found in the membrane of endoplasmic reticulum. Then students come to the functions of endoplasmic reticulum. First one is intracellular impulse conduction. The impulses they travel biochemically through ions, and there is a conduction of impulse in the cell, inside the cell also, and this is done by endoplasmic reticulum. How does it perform the function of impulse conduction? It does by presence of ionic gradient and electric potential on endoplasmic membrane. The sarcoplasmic reticulum, which is the endoplasmic reticulum of muscle cells, acts as intracellular conducting system. Transmit impulse from sarcolemma, which is the plasma membrane of sarcoplasmic reticulum muscle cell, into deeper regions of muscle fibers. Then the endoplasmic reticulum is also involved in intracellular transport. The endoplasmic reticulum creates a cellular circulatory system, and it is involved in import and export. Intracellular circulation of genetic material and its transport is effected by endoplasmic reticulum. Many places ATP synthesis is done by the endoplasmic reticulum, so this is one of its function. Then protein synthesis, as I've told you that the surface of rough endoplasmic reticulum is laced with countless number of ribosomes, and these ribosomes, they prepare proteins. So endoplasmic reticulum in that sense directly doesn't involve in protein synthesis, but provide surface for attachment of the ribosomes which are involved in synthesis of proteins. Then lipid synthesis, as I have told you that the smooth endoplasmic reticulums, they are devoid of ribosomes, so they are not involved in formation of proteins. However, they are involved in formation of the lipids. In Golgi bodies, glyceride get associated with the protein synthesized by rough endoplasmic reticulum to form lipoproteins, and the tubules synthesize steroids which as cholesterols. Then glycogen metabolism. The smooth endoplasmic reticulum withdraws glucose from glycogen by action of glucose 6-phosphatase, meaning it is involved in glycogenolysis, which is breakdown of glycogen into glucose. Another important function which is performed by endoplasmic reticulum is it provides the mechanical support to the matrix of the cell, and therefore it acts as cytoskeleton. The smooth endoplasmic reticulum, as I have told you earlier, is involved in cellular detoxification. That means it neutralizes the toxic elements which are byproducts of the metabolism and catabolism happening in the cell all through the day and night. Then another function is the maintenance of osmotic pressure within the cells and maintenance of ionic gradient. As in sarcoplasmic reticulum, when muscle gets stimulated, calcium ions are released. The differentiation of Golgi and nuclear membrane takes place from endoplasmic membrane itself. Then formation of microbodies is done by endoplasmic reticulum. They are formed as dilations of an ER rich in enzyme peroxidase, hence called peroxisomes. So peroxisomes are differentiated from endoplasmic reticulum, and such vesicles, they are rich in peroxidase enzymes. Then the endoplasmic reticulum is involved in synthesis of glycerides as phospholipids, triglycerides and glycolipids, along with alpha ascorbic acid, also synthesized in endoplasmic reticulums, and other chemical reactions which takes place in endoplasmic reticulums are deamination, hydroxylations, oxidations and aromatizations. As far as the genesis of endoplasmic reticulums, two beliefs they prevail. One from nuclear membrane. As the membrane of endoplasmic reticulum resembles nuclear membrane, it is widely believed that endoplasmic reticulum develops by epigenation of nuclear membrane. This belief is buttressed by the fact that nuclear envelope is formed from vesicles of endoplasmic reticulum at telophase of cell division. Then another belief is that they develop or originate from annulate laminae through multi-step mechanisms, and it is believed these days that ER membrane are synthesized in annulate lamellae, which are double membrane sheets 2 to 12 arranged in forms of stakes arising from nuclear membrane. The outer nuclear envelope forms finger-like processes which are pinched off into cytoplasm in form of vesicles called as blebs, and this phenomena is called as blabbing, and it is through blabbing of annulate lamellae that the biogenesis of endoplasmic reticulum happens. So students, this is all in this lecture. We'll continue in the next lecture regarding the endoplasmic reticulum. Thank you very much.